This is Laverstow Park. What's the story in England with the bunny rabbits that flew a rebellion? Remember that classic literature? Oh well, this is where that story is set. So, all of these fields are a part of the farm. So the local town, so we're dealing with Overton here. So working with the guys, with the farmer here. Which fields are you going to sample? I initially started out saying, well, sample every one. So they said, no, can't afford it. So let's pick the representative. And so from their arable systems, so we're looking at vegetable production in here. So 3 of their typical fields. And so we're using them as examples of all the rest of their vegetable production systems. How are we going to get the biology in there? So where did we start from? So 5 samples taken from the plant that we're concerned about. So in the first year, I believe this was carrots to start with. First year, this is lucerne and cabbage. And the second year was something else, third year was something else, but the rotation eventually came around. So we have all of that data of how the biology has improved by putting on compost tea. And then there are grapevines. Yes, they grow grapes in England. Southern part of England, so you can't call it champagne. It's sparkling wine. That's the French. So 3 of their areas with grapevines or hops, so comparing those, and then 3 representative pastures. So going into each of those areas, predetermine where you're going to take your samples. So we typically look at where are the weedy areas, and we would have a different sample from where we've got high weed problems. Or an area that's real wet and low, we would be taking samples, 3 samples, 5 samples from there. The top of the ridge. So we have to start thinking about all of these variations that we must pay attention to. When you deal with a lot of soils people, they will say, you just take random samples and you pay no attention to what the plants are telling you. Now, you start taking samples like that, and we're going to go over the bare spots, we're going to go through the buffalo grass, we're going to be going through the dandelions, we're going through the area where we got some shrub growing, and taking samples and consolidating, mixing all those together. What are you going to learn about? An average of all of those different kinds of plants, well, they're massively different. They have the requirements of bacteria fungi. So we're not learning what we need to learn about when we're sampling in this fashion. So we are going to stratify. Let's take a look at that field and the fact we've actually got the top of a ridge. The soil at the top of the ridge is only this thick. As you go down to the pond, the soil, that A horizon, is this thick. Very different things going to be going on there. You don't want to be taking a sample from here and mixing it with a sample from here, because what are you going to learn about? Neither area. We really have to be taking samples from along the ridge line and figure out what's the biology in the soil, how we have to manage that, as opposed to down here where it's really wet. And that soil stays wet the whole growing season. We're going to do different things here to fix the different problems. Well, we got that weed patch. Why do we have a weed patch there? So let's sample in that weed patch and sample within that weed patch. That's better?
Ooh, look at this. This is fun.
Keep going in there.
On the neck.
This thing kills us. Put it on your neck. Oh no.
I'll wait. Rawhide. Okay, this is gonna be fun. Every time I have to sniff, you're gonna be, and he's
Save your
taking
voice, save your voice,
pictures of it.
Right? Got it all on video.
Yeah.
I'm gonna save your voice.
Yeah, that's a good idea. I can still talk come Friday. And so like even within the week time, we're going to go to the plant we care about. So we could learn what's going on with the plant we care about, and then we might be sampling from one of the weeds that you really don't want to have in there. What's allowing that weed to win against the crop that we do want? How are we going to manage that biology in that soil?
Right there.
That's fine.
Is it better? You guys?
Is that better? You can hear me? And I don't have to talk as loud. That's good. The volume. Okay, very good. Okay, so job one is when we start working with people, it's always where are you going to get your compost from? How do you know if it's any good? Start looking at all the compost available to you. Is anybody in this vicinity making compost that has the biology in it that you need? Are you going to buy thermal compost or worm compost? I don't care. Whichever one has the biology that you need. So you want to be looking at your soils, deciding what do I have to have in the compost I'm going to buy? Or maybe I have to make it myself. Especially when you're buying compost, it's expensive or it can be expensive. So there's where most people are going to be making a tea or an extract from that material so that you're putting out the biology. And then monitor that as time goes by, the biology remains in the compost. You have to store your compost correctly, or you can wipe out the real good organisms that are in the compost. So we want it to stay moist, 50% moisture, no less than 30% moisture when we're storing compost. We want to make sure that it stays aerobic. The organisms keep cooking along in that compost. You don't really have to feed it all the time. You might want to feed it just a little bit of fungal food if you're losing the fungi over time. So we'll talk about those things when the day we'll talk about compost. How much compost do you need? Just the barest inoculum. We're adding the inoculum into the soil. You don't need to be putting out tons. Drives me crazy when somebody says, I put out 10 tons per acre. Jeez, you're rich. Or you're making your own compost. It may depend on the toxic material that's actually in your soil. The more toxics that are present, the more compost you may actually have to use, because the compost is food to feed these organisms. One of the things we'll talk about again a little bit later is this question of where do you get really good food to feed your fungi? And it is the humic acids. They're really good fungal foods. This is what feeds the good guys. Humic acids. It is a material that causes the 70% cocoa color in your compost. So if we want to obtain humic acid, you go to the store and you start looking at the humic acid that's available in a garden store, a nursery, or someplace like that, and you'll notice that that humic acid that they sell in bottles, nicely sealed up, comes from leonardite. Leonardite is a soft brown coal. It's on its way to becoming nice black coal that's got some BTUs in it, but it's not there yet. So it needs another 100,000 million years, and it'll become black coal. But it is from plant material. All coal was once upon a time plant material. It's just in the process of becoming crystallized. So what they do is they mine the leonardite out, soft brown coal, And they'll go through an extraction process. And the first step is to typically add that crushed up leonardite to the strongest sulfuric acid or hydrochloric acid, really nasty stuff. And that causes all of your fulvic acids to drop out of solution. So they collect the fulvic acids. Except look at what they use to extract. It's taken all the water out of this material. You denatured your fulvic acid. It's not usable by any microorganism, and it won't become usable by any microorganism for perhaps years. They then take the leftovers from this material, and they're going to add in like 90% potassium hydroxide, really strong base. And the humic acids drop out of solution when you're in strong base. So now they can collect the humic acids. Well, yeah, it is humic acid, but there is no fungus on this planet that can use this stuff until they've chewed on it in the wild and renatured it. So if you're buying something that is an extract from leonardite, don't plan on the bacteria or the fungi or any microorganisms being able to use this stuff for a couple of years. Don't put this in your compost tea, because you're not going to grow anything. You're probably going to kill your organisms if you use this in a compost tea or a compost extract, and certainly if you put that into your compost, you're causing more damage than you are benefit. So where are you going to get your humic acid from? Well, if you're making compost, you're already making humic acid. The brown color, that nice dark brown 70% color in your compost can easily be extracted. So take your finished compost and put it in a nice sieve. So your compost goes in here on your sieve, and so the compost sitting in there, you're going to just add water. And you add enough water, passively moves through your compost, starts dripping out the bottom, and you collect that water that's moved passively through your compost. And what you'll see is you have a nice dark brown 70% cocoa color if you've had good biological activity in that compost. So here is a way to tell whether you've got good compost or not. Run some water through it. If you've got a nice dark brown color and that water coming through, you have to have had good fungal growth, or you wouldn't have made humic acid. And this is water soluble. Now when you need to grow some fungi, use this material. It doesn't really have many bacteria, it doesn't have any fungi, protozoa, nematodes in here typically, because it's passive movement of the water. When you passively just by gravity let this move down through, that's not enough of a washing action. You're not going to extract any of the organisms as that moves through your compost. So you don't have bacteria, fungi, protozoa, or nematodes coming out the bottom. This is pretty much a sterile liquid. Because bacteria and fungi, do they want to wash out of that compost? Do they want to wash away from their source of food? No way, Jose. No, don't take me away from my food, no! So they have the little voices screaming, and you can't get them off without really shaking and moving and trying to rip those organisms off those surfaces. So you're making compost. Extract, you get your own humic acid. How expensive is this stuff?
Great.
It's the cost of the water. A little bit of your time. How expensive is that stuff extracted from leonardite? Jeez. $70 per gallon? Wow, painful. And it's getting more expensive all the time. So I want to encourage you to stop using the leonardite that they sell in the store that's extracted. The leonardite that's been extracted to produce the humics or the fulvics, just not worth your time and effort. Make some good compost, make your own, cheap. Questions?
How do you see that? I mean, what kind of screen do you put on the bottom?
Just a typical zip, you could even use a colander that you do your pasta in.
Oh.
Doesn't need to be any finer mesh than that. If you've got a compost that's, got a lot of
Like the screen, can you use some of that, the screen would be all right?
Yeah, and we might have some fun trying that with some of the compost that people bring in to look at. In the microscope, we could try making some humic acid, so bring a little bit extra. Push it back there.
The drippings of your like a worm house or something, is that your humic acid as well?
When you're looking at a worm bin, make sure that it's not dripping all the time.
Right.
Because if that's water saturated worm compost, well, you ain't got no worms in there anymore, sorry.
Right.
Oxygen does not move through your water very well, it's gone anaerobic, your worms are strict aerobes. But if you, on occasion, pour water and let that water passively move through and collect that material, that's some of the best humic acid you want to talk about, because with the worms, it's probably also going to come with some plant growth hormones. It's going to come with some really nice enzymes that are good at decomposing other materials, and you're leaching out those nutrients, those enzymes as well. So you can get some amazing plant growth if you put worm leachate. It's not a worm tea, don't call it that, because tea has got to have organisms growing in it. And we don't want any organisms coming out of your worm compost or out of your compost extracted like that. We want the humic acid here. So we can use it to grow other things. But it is worm leachate is wonderful stuff. It has a great effect on plants, but a short-term effect. Once those enzymes are used up, you're not going to get any more additional benefit to it. Once that humic acid's used up, you're not going to get any more benefit from it. So do you use the leachate coming out of here, or do you use a compost? Well, if it's just food resource, leachate. If you really need to add the inoculum of the beneficial organisms, you're using that compost, use extract, compost tea. Comes with the biology.
Mine was a similar question about the leachate. How do you apply that? Would you dilute it? Would you just apply it straight?
And again, it's going to depend on what you have to do to the soil. So if you've looked in your pot and you can see that you've got lots of bacteria but no fungi, you probably want to add this just straight, just water with humic acid for a couple days. If you're putting it on just to kind of keep the organisms happy and going, you might dilute that 1 to 10, 1 to 50, just to kind of keep things going in there, where you're not really trying to grow massive numbers of organisms. When we're dealing with soils that have a lot of heavy metals, humic acid will tie up any salt. It will tie up any heavy metals. And so if you have toxic materials in your soil like that, you want to try to tie them up, humic acid will do a great job. Just remember that if you don't have the biology, humic acid will only tie up a heavy metal or a salt for about 2 weeks, and then it's released again. So if you put humic acid in, you better be putting biology. So then you can take those salts, those heavy metals, and have the organisms put it into the structure of the organic matter. Once the microorganisms tie up those heavy metals, once they put them into the structure of the organic matter, the half-life on those toxic materials is about 500 years. They don't come back out. They are not going to be available to your plant in your lifetime, unless you're planning on living 500 years. So I think we're safe there. But you have to have the biology to take those heavy metals using the humic acid to put it into the structure of your organic matter.
With the worm leachate or the humic acid, is there a way to store it? It needs to be oxygenated or it can be sealed?
It can be sealed, so as long as you haven't really extracted any of the organisms. And so how do you know whether you've got biology in the extract?
Microscope.
Bingo, microscope, you're learning, aren't you? The choices are it depends or microscope. So look at it. If you don't have any, you don't have very many organisms at all, seal it up. That's one way we always test our extract, to make sure that we really don't have any organisms in it, is to actually put it in a bottle, seal it up. Because if you had any organism growth on that humic acid, what's happening to your bottle? It's really fun when it explodes all over the inside of your garden shed. Oops, we messed up on that one. So don't be pushing the water through, just passive movement. Let it soak on through. You may, put out a little bit of water on, a little bit more, a little bit more, a little bit more. How much water should you put on a good compost like that? You take that first amount of water, nice, rich, dark brown color, wonderful, really good. Now let's put another container and capture some more. How many cups of water can we put through a cup of a good compost? And what we find is that if we've got really good humic acid that's been growing in our compost, we can run 12 cups of water through 1 cup of compost. And we will continue to make really good compost tea until it starts getting too light. So, did we have some really good fungi growing in that stuff? Yeah. Can we make compost at Rodale? Oh yeah. Question here?
I didn't want to ask a question, but I wanted to address David's question earlier about how you screen and sieve. I found a really cheap and effective solution is that at most paint supply stores, they sell 5-gallon strainers for paint buckets, and they're cheap. They cost under $5 for a package of 3.
Paint screen.
Strainers.
Strainers.
It's just a nylon kind of straining mesh that fits into a 5-gallon bucket
Yeah.
for straining paint, and they work excellent.
They got the solid ones too that sit on the rim. Those are really nice.
What, like Ace?
They got on sunshine.
HB?
Yeah. Ace.
So whatever works. Just don't let that bag hang down in the water, because then the organisms in the compost start growing, and you're going to go anaerobic really fast.
I'm a little confused because if your garden's mostly organic matter, compost, and it rains, aren't you losing all the humic?
You could well be having some of that material leach out. Yeah.
But
So
you said you had the organisms, you weren't going to leach it out, so I don't know how can that.
you aren't going to leach into your soluble inorganic nutrients, but some of that soluble organic material may need moving from your compost. Like if you have a bed at Rodale, one of the tricks we like to do a lot of times is where we've got really horrible soil where we're dealing with somebody, we'll put in an 8-inch bed wood, and then we start growing right in there. Okay, so now every time it rains through that compost, we're going to be leaching humic acid into the soil below that bed. That's exactly what we want to have happen. Because we're putting the foods to hopefully grow some good biology below that bed, so someday we can take that away and we can start growing really good plant material straight into that soil. So it's a good thing that happened. And this stage is going to be a lot. Yeah, that's how far down are the roots of your plants going into the cracks and crevices of the lava. And maybe we need to be feeding them. And so I think it would be a good way to feed your root systems through the cracks and crevices of the lava. Let those organisms keep chewing out the lava so someday it can become soil.
Can I clarify for myself? So humic acid is just something we're adding to our tea that kind of boosts or wakes up our biology.
Yeah, more of a fungal component. It can be woken up typically.
So it wakes up your fungal biology.
The fulvics are typically kind of intermediate. Some of the fungi and some of the bacteria wake up with the fulvics. Now we're probably also leaching out some oleic acids, some carbohydrates, some proteins. So exactly what is the mix of all those organisms in your compost? Look at the biology that grows. And then you know what were the food sources that you leached out of that compost. Those are minerals.
Does that process have anything to do with biochar?
She said that word. Sorry. Biochar I find kind of challenging right now because there's just so much hype I don't know how much people here listen to the biochar hype, and it just frightens me when somebody, this is the solution to every problem on the planet. Right. Yeah, not possible. Now, biochar, if you make it out of good plant material, and you're really careful about the temperatures and the oxygen concentration, you can stabilize a lot of carbon that way. But exactly what's in that carbon? So, biochar, what are we really talking about here? When you take any material that has a lot of carbon in it, and we subject it to really high temperatures, in really low oxygen conditions, we will turn that into charcoal. And that's what biochar is. There is no biology in it. Sorry when they say, good microorganisms in here. No, because how do they survive 6,000 degrees Celsius or whatever it is, Fahrenheit, whatever. How are they surviving 1% oxygen when you should be up around 22% oxygen? So biochar, it's an inert material. It has lots of spaces, lots of cracks and crevices, and it's kind of condominium housing for bacteria and fungi. But do we not have other things in soil that we already have condominium housing for bacteria and fungi? Yeah, it's called clay. It's called aggregates. It's called. So, under what conditions do we see a benefit of adding stabilized crystalline carbon into the soil?
It hasn't got any of the humic acids that
Now, all the humics have been burned off. They've gone off as gas as you char that material at that extremely high temperature and really low oxygen levels. So we've got a biochar experiment going on at Rodale. Somebody came and said, we want you to test our biochar. And so we said, well, okay, we've got a not organic field out the back there where our neighbors may let us use that field. So we have a certain pattern that we put biochar here and here and here and here and not biochar and have put in the crop. First year that we did this, absolutely no difference between where the biochar was and where the biochar was not. Now we're repeating the work, so we're going to observe and see what happens the second year, because maybe it takes a year for the biology to get up and going on this. So stay tuned. It'll be on the Rodale website. Take a look. Oh, well, let's see. It's a real heavy clay soil that we put that biochar in. So think about clay. There's already condominium housing for our microorganisms. There's already lots of food for the microorganisms in that soil. So, yeah, is it really going to bend? But what if you put that biochar into pure sand? So now, let's have these guys be honest about where it could be a benefit. If you're putting biochar into clay, I don't think so. Worthless, pointless. Yeah, it was a waste of money.
If you're in Africa and they have low rainfall.
Yeah, now I see where maybe it could work. Could we have some data? I am a data-driven kind of person. I must have data. So until they show me some data. Now, we worked with some of the first people that were, the lead people on the whole biochar craze. And I would invite you to go and take a look at their, make me laugh, scientific experiments. So they have pictures of this agricultural field where they put the biochar on it. And take a look at that agricultural field. The corn grew about this high. Have you ever seen an agricultural field where you're using pesticides and inorganic fertilizers where the corn only grew this high? That's not an agricultural field. So don't pretend to me that they're comparing what the biochar does with agriculture. They went into a wheat field and put some corn seed out there. It has nothing to do with agriculture. And then over here they put the biochar into the soil and yes, the corn's growing this tall. No corn, no ears of corn on it. But then they're all like, wow, look at how much better the corn grew. This is fantastic. Biochar is the the cure for global climate change. Where's the data? Show me the data, because did not impress me at all. But they hyped it. Did they get some backers for that biochar? And look at the hype that's out there. There was a biochar convention in Rio de Janeiro 2 years ago, almost 2 years ago now. And as you went around to all of the biochar booths and collected the biochar from each one of those places, and then it was analyzed using mass spec, looking at all the toxic chemicals that might be in that material. 80% of the biochar being sold at that conference was so toxic that if you put that around your plant, you would kill your plant.
Just because of the woods that they're using, or?
Whatever material they're putting in there, but we got people making biochar from biosolids. They're making biochar from whatever mix of toxic material you want to talk about. They're making biochar from it and now saying, see, this is wonderful stuff. Put that into your soil, kill your plants. Is there some hype? So when are we going to have regulations to prevent that kind of garbage being sold?
Since you're on the topic, fish hydrolysis, that seems to be a rage too.
Look out. What is that? What is fish hydrolysis? Well, it's a pretty good fungal food. If you're actually getting real fish hydrolysis, so when we're making compost tea, when we're making compost, and you want to give the fungi a bit of kick in the butt. Fish hydro, meaning water. Lysate, meaning it's all broken up. It's been pulled apart. So we're using water to break our fish up. So it's going to contain everything that's in the fish. So on a small scale, take your Waring blender. Buttons. Nice cute little blades in the bottom. Put a little bit of water in your Waring blender. And now let's drop our fish in there. Turn the button on. Fish hydrolysis. That's what's meant. Fish hydrolysis. Don't like the bones in there? Well, just blend it up a little bit more. Don't like the scales? Just blend it up a little bit more. So we got some good calcium in there. We got all those guts and all the food that the fish might have been eating. Well, really. We're going to take the fillets off the fish first. So human beings are going to eat the fillets. And that's the remnant of the fish. So it's all the oils. It's all those good nitrogen, protein, some pretty complex proteins, oils, great fungal foods. Absolutely no heat can be involved in this. Making fish hydrolysis is a cold process. As soon as you start heating up that fish hydrolysis, you will cause, there's the flame, does that look like the top of your, here's my container with my fish hydrolysis. As I heat this, it causes the oil to separate. And now this is no longer fish hydrolysis, this is a fish emulsion. Why would you separate the oil from your fish? Because this is really expensive stuff. If we can take one fish, we're going to get a couple microliters of chondroitin in that oil. And so the guy's taking that fish material, they'll blend it up nice and fine, but then they're going to heat it in order to be able to extract that chondroitin. 5 microliters of chondroitin is worth about $5,000. People use it for muscle, tissue damage. So anyone who has joint problems, guess what the active ingredient is in those salves and those creams that you put on your joints? Chondroitin. This is expensive stuff. So they all say, oh no, we haven't heated it up at all. Oh no. Yes, you have. Because take a look at the bottle when it comes to you. If that bottle of fish hydrolysate separates, and now you've got a layer of all the fish proteins and meat and stuff, and there you've got your oil sitting on top, this material is fish emulsion, and it's not gonna grow any fungi at all. It's gonna grow bacteria, not a fungal food. So be real careful on the fish hydrolysates that you buy. Yep, it's separated the amount, and now that's no longer a fungal food typically because they've removed most of the really good fungal foods. So recognize too, this is fish hydrolysate, and when you put it into a container like this, how do you prevent microorganisms from growing? You're going to have to put a preservative in here. And typically, what is the preservative? So preservative. Oh, it's something. Typically, it's going to be phosphoric acid.
Is that what prevents it from smelling too?
If the phosphoric acid didn't do a good job of preventing microbial growth, and you open this thing up and it's like, grow up. Then you've got microbial growth in that material, and now what kind of microorganisms are growing? Take a look, microscope. And what's the probability that if it stinks to high heaven, aerobic, anaerobic? Where's your nitrogen gone? Yeah, so as you open this up and you smell the stink, okay, not good stuff.
What about like homemade teas like that? Like in natural farming, like fish amino acid, where you're taking fish, you know, the leftovers of the fish, and you're putting it in a container and covering it and letting it sit for 6 months. It ferments and it stays smelling good because you've added some, you know.
If you put microorganisms in there that prevent the anaerobic stinky smells, then okay. So typically lactobacillus again.
Right, or microorganisms that you collected with your rice, and it stays smelling fine. There's no stink, and then in 6 months you collect it, and it's
I'd still want to look at it for the microorganisms. Have I actually grown some good fungi, or is it just strictly bacterial? What kind of protozoa are in there?
So look at it anyway.
Yep, and then you know what's happened to it.
Yeah.
Is it worth putting on my plant, or is it going to kill my plant? Be looking for the ciliates. If we don't have ciliates in there, then you're probably pretty safe using that as a food source. But what is that material actually going to grow? Is it going to grow bacteria, or is it going to grow fungi? So add some of that into your compost. Maybe a small amount of your compost, you know, so take a nice container, you know, some kind of box or something, and then add a little bit of that material in there. Have a control, where it's just your compost and you add water. And then another container where you've got the compost and now you add that material into it. And now look at the difference. What did that actually grow? Wow, I got a billion bacteria! And they're fungi. Oops, not what you wanted to see. Or you go, when you're comparing these two, it's like, okay, the bacteria is still there. Like, man, look at fungi, wow. You open the box and you go, wow, look at all those beautiful, thick strands of fungal hyphae all over everything. And then you know, what is that feeding? So variation, time to time, when you're dealing with those sorts of materials, please recognize that every different kind of fish has a different set of oils and a different set of proteins. So as soon as you got, you know, this time you made that material with marlin, and next time you make it with ihahi, or you make it with, are you going to have different results? Good. So you get some work, don't you? You all get, see why it would be useful to work together? So every time one of you is testing something and you go, oh, this did not work, wouldn't it be nice to share with the whole group? So David, put you together as the center point for collecting all this kind of material and then putting out the list of what doesn't work and what does. What does this grow? What does Tabasco sauce grow? There's some really good bacteria. Test it. What's it actually doing? So sampling compost. We sample compost when it's mature. And again, we want to reach into our piles at a typical depth and make sure it's at a typical moisture level. So you can put your arm in, dig in about 2 feet. So the depth of your arm is about 2 feet. Grab a sample. As you're pulling that compost out of that pile, you're checking for moisture. So if I reach in there and I pull a sample out, and while I'm pulling that out, it's already starting to go drip, 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 drip. 100% water saturated. It is anaerobic. Just put it back. Walk away, and hopefully the stink will not have transferred to your hand. Yeah, when I met Prince Philip, I had to go behind. It's like, nice to meet you. Because wow, I've been having fun with the compost piles the day before, and I can guarantee you that the Royal College does not know how to make compost. How was our compost? We really need to send people over training. So, reach into that compost pile. If it's not going drip, 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 this is a good sign. So now you start squeezing it. So enough compost material that it fits nicely into your hand, squeeze it. If we're at 50% moisture, we're going to just get water in between our fingers. Or maybe 1, no more than 2 drops. What if I get 10 drops out? Okay, now you're up there around 60% moisture. But we want our compost piles about 50%. So if we're getting water between our fingers, a couple drops coming out, we're at 50% moisture. And now this is significant, available to your organism's water. Don't go with gravimetric moisture when you're dealing with organic matter. It really doesn't have any meaning whatsoever. We want to know what's available, and you get this by squeezing. So 50%, 1, maybe 2 drops of water.
What do you mean with gravimetric?
Gravimetric moisture is, scientists who want to do everything so we can get a number to the 10th decimal place. Gravimetric water. You're going to take a little pan and you're going to weigh it. And now you're going to add add your sample to that pan. So I put some of my compost in there, and I'm going to weigh it again. So I know the weight of the fresh material with the water. So there, you've got your water in there. So now I take the pan and I put it in the oven, typically at about 80 degrees Fahrenheit overnight. And what you'll notice is in the scientific literature, we have argued about this number for years. We used to do this up around 150 degrees. Well, you blow off some of your carbon, you blow off some of your nitrogen if you're getting it too hot. So for a number of years, the number we used was 105 degrees Fahrenheit. Well, you're still blowing off some of your carbon. So today, most people use 80 degrees to dry it overnight in the oven. So 18 to 24 hours. We're trying to do the standard here. And then you re-weigh. So now you have the weight of your sample weight. Mental blankness. Love it. Weight of your sample dry. So the difference between your fresh and your dry weight is the amount of water that you had in that amount of fresh material. So do the calculation, how much water per gram. And now grams of water, divided by grams of sample. And we can calculate percent moisture, gravimetric moisture.
That doesn't work.
Pretty meaningless when it comes to how much water is available for your organisms or for your plant. So this is better when we're really trying to figure out if we have adequate moisture to grow your organisms. So you reach in there, 1 or 2 drops, great, life is wonderful. But what if you squeeze that and no water comes out? You're less than 50% moisture. So no water coming out when you squeeze. We're less than 50% moisture. So now we want to open up our hand. Open it up. If that material sticks together, if it stays as a nice clump in my hand, then we're about 40% moisture. So it stays clumped. If when I open my hand up, it falls apart in my hand, those bits and pieces just fall apart, they don't stay together, we're at about 30% moisture. So it doesn't stay together. And then what we really want to do is apply the blow test. Do a blow job. So you've opened up your hand, it's kind of falling apart, you go, and if a whole cloud of dust flows that way, you're at less than 20% moisture. Your organisms now officially hate you. And they will not wake up. They are going to give you a whole bunch of trouble. It's difficult to get them to come back to life once you let your compost or your soil get down that dry. You're below wilting point, life is not good. Microorganisms, when they start getting dry, they will put out layers of hydrophobic material. Hydrophobic. It's not going to wet up. The reason the organisms put out that hydrophobic layer is as things get real dry around them, they're wanting to keep all the moisture and prevent it from drying up and leaving them without any water. So they're going to put out this layer of hydrophobic material. And of course, if you keep drying them, they're going to die, but the hydrophobic material is left. And once that hydrophobic material is left, it's very difficult to wet your soil back up or wet your compost back up. Yeah, I don't know how many of you, like on citrus, have you ever noticed where you're watering your soil and you've got this patch where you cannot get water to go in there? It just runs off, hydrophobic. It's because you allow that soil to get too dry. The organisms have made these hydrophobic materials and it's virtually impossible to get that material to wet back up. Don't let it happen. If you possibly can manage it, you don't ever let your compost get that dry. So keep it above so that when you squeeze that material, at least some of it holds together and it doesn't blow off as dust. So when we're measuring moisture, use your hand. So going into our compost pile, take a sample about 2 feet in, measure the moisture as you're picking it up, that handful goes into your bag. Walk around your pile, take the second sample, walk around your pile, take a third sample, maybe 5. So there's your compost sample. You've gotten it from a number of places in that compost pile, so it's an average of all of that. So now we're going to go look at this material on a microscope and find out what biology is. What's going on with the compost over time? Is it getting better? Is it drying out? So we maintain the conditions in that compost pile so the organisms will stay alive. How much compost? Well, here's a it depends answer. 1 to 10 tons per acre. Well, 1 ton if you really have pretty good biology in that compost. 10 tons if you don't have much biology at all. Well, is it really compost if we don't have any good biology? So folio applications, we talked about those. So 10 to 15 pounds of compost in 500 gallons. And we're applying that 1 to 5. If we got good biology, 1 gallon per acre. If it's okay, pretty good biology, 5 gallons. If it's really pretty pitiful biology, well, you better use 20 gallons per acre. If you really don't have a good biology in there, don't bother. What's the most expensive part of putting compost tea out? It's certainly not making the compost, and it's certainly not turning that compost into liquid form. It's the spraying. There's the time and the expense. And if you don't have the biology in that compost tea, don't bother. You're wasting a lot of time and effort applying water. When's the last time you noticed putting water on your plants got rid of the mildew? Yeah, so don't do it. Seed dressing, so with the seed dressing, we just want to get that biology up as high as we possibly can. So you're going to make a good compost tea. Extracts, again, it depends on the organisms, how much compost you're going to use. So be looking at your biology. Determining success when we apply these things out there. You monitor your soil life. Is it getting balanced where it needs to be? Get back out there with the penetrometer. Have we broken up the compaction layer or not? Do we need to put more compost in there? Do we need to put more tea? Let's be watching our root depth. Are our roots just sticking up here, or are they starting to grow down? So you might want to have a soil pit. You might want to be going out and digging into your soil. Are your weeds still there? Well, if your weeds are still there, what's that telling you about your soil? You haven't fixed a thing. Get to work. Now, if your weeds are disappearing, if your weeds are the things that are sick and unhealthy and unhappy, great. That's exactly the right thing. Are you seeing the disease disappear? Insect pests, yellowing, burnt borders of your leaves, that's inadequate nutrition. So if your leaves are heavy metals, how many of you have seen heavy metal damage on a fruit tree? Your leaf bubbles. So it's got a real misshapen and funky appearance to it, heavy metal problem. My yard, when I bought my yard back in Oregon, I did not realize at the time I bought it that they had put black plastic in the soil about that deep in underneath the sod. They had put in black plastic. So I went in, I'm going to plant some fruit trees. Go in there, put my shovel into the sod, and what the heck is that? Now I can't go any further. Start digging it up. Black plastic. That far, that deep, as a weed barrier. Yeah, well, that worked. Not. And so I started ripping, dig it all up, start ripping up the black plastic. And the color of the soil underneath that black plastic was the color of your blue shirt. So anaerobic that all the copper in that soil was so anaerobic that I was looking at anaerobic copper in that soil. Great, what does that do to your plants? My poor trees. For the first couple of years that I was dealing with my trees, all of their leaves were just gnarly. Could not eat the fruit off that tree. Now, when I did pull the black plastic up, got oxygen going back into the soil, that copper immediately faded. The soil faded in about a day or two. And then I could dig down and find blue soil again and let it aerate out a little bit more. So I had a lot of work to do. And so looking at the effect of that heavy metal on those fruit trees, it's not until you see that bubbling disappear, that really odd misshapen leaf material. You have not dealt with that heavy metal. I had to get the biology back in there, I had to get good organic matter, so it took me about 6 years to bring that back to life. 6 years before I could make crab apple jelly, before I could eat the cherries off my tree, before I could make cherry pie. But if you come by my house in the springtime, you can enjoy all the cherries that we have. Now, and I've got 7 different types of apples, and I've got blueberries and raspberries, and edible gardening every place. I like to go outside and graze about this time of year. Corvallis. I mean, of course, I'm not in Oregon in the summertime anymore. So my husband gets to eat it all. And you know what? He needs help. We'll be there. Go for it. Yield, number of food, tiller, so all of the things that you could measure. How much less water you have to use now as compared to before. And of course, you don't need fertilizers and your plants are growing really well. And you're not out there pruning all the time, and you're not out there putting on herbicides and all the benefits that you can be measuring. Which of the biology is most important? Scientists ask you these kinds of things all the time. In your food web, which is the organism that's most important? All of you. It's like asking somebody, what's the most important organ in your body? Right? Not scientists just have to let them. What's the most important organ in your body? If you're a scientist, what's your answer going to be? My brain! This is the most important organ, right? If you're romantic, what's your response? My heart. Okay, I'll let you have both your heart and your brain, okay? You're going to give me all the rest of your organs. How long are you staying alive? Just try to exist without your stomach, or your lungs, or your spleen, or your skin. Think of that. It's a holistic system, just like your body is. All of the parts have to work together. So there is no most important. It's all of them working together. You can't retain nutrients without the bacteria and fungi. You can't build structure without your bacteria. And then your fungi. You're not going to make plant-available forms of nutrients without the protozoa, nematodes, microarthropods. One or all or some combination. It's the whole food web that we have to understand. So, we've got 45 minutes to keep going, so let's do a little question and answer, and then we'll go on to lessons Elaine learned along the way.
So here we have a real problem in our pastures. The firewood that's the field name. It's I guess sourdough or something like that.
Alien invader.
Yes, it is actually. It came in just and it's taking over the whole island. It's toxic to the grazing animals, and so the rangers really don't know what they're going to do about it. I mean, I suggested that we try to do something with the soil. I don't know. Is it like, okay, get great money and try to look at it?
Yeah. And so the step one on something like that is to go out into the fields and find out what the biology is in the root system of the fireweed. It's a strongly bacterial dominated plant in my experience. How are we going to select against it? Well, let's get the fungi back into that system, and places where we have worked with it in South Africa, we've gotten rid of it completely. Working with ZZ2 in Limpopo province in South Africa, they've gotten rid of that fireweed out of their pasture systems. Not a problem. So where I've dealt with that or similar kinds of alien invader plant species, it's fairly easy to get rid of it. We need to have replicated scientific trials done. When I work with growers, we'll typically go out and we'll take one small area because you got to learn how to make compost. And then how are you going to apply it? What's the easiest way? And people usually mess around with that for a year before they really get this under their belt. And then they go out and they're applying it, and all of a sudden it's like, wow, this is so easy. And the next thing I know, the next time I come back to look at the trial as it's going on, walk in the door and you go, what have you been doing? You've been putting compost tea everywhere. Because what's a grower's concern? Money. Making the whole farm productive with the least effort for themselves. So there goes my scientific experiment. Thanks. And also once again, it goes down. So see why the reason I'm at rodeo is because I'm in control of the fields. Somebody goes out there and puts compost tea or compost or whatever the treatment is. in the wrong place, I will kill them. So, when you find dead bodies along the side of the road, go down, yes, they screwed up. So, learning with growers, they're not interested in replicated science, they're interested in making things work on their property, and we have a million examples of that.
Just a question, because you're talking about everything's out of spot. Where you make your compost, is that important too? For example, you were talking about the fruit trees where you wanted small cover crops. Would you then suggest that if you have an acre patch of that somewhere on your property, that you build your compost pile on top of that, or?
It's certainly a good way to get rid of weeds, is build your compost pile on top of it, because all the leachate going through is going to be pushing that soil along in a direction that's going to wipe out your weeds.
But for example, like you can find spots that don't have big invasive grasses like there. So there's something in there on the bottom already that's keeping those invasive grasses from just sprouting up in those spots. Would you look for that?
Probably not.
Okay.
Because we've got really good biology in the soil and we're growing a plant that's not the invasive species, I want to kind of keep that safe and let's go put our compost on top of the invasive species, because we know that we're going to be putting all this good biology, exudates, humates into that soil, and so we're going to be shifting that. We're going to kill it because there's no sunlight in the plant too. So I tend to compost on soil. Small scale, it doesn't really make much difference where exactly you choose to compost. So for example, with Nona, I was suggesting to her that the place to compost is right underneath the trees. So within the drip line, small size, because here in the tropics, you don't have to worry about cold temperatures at night. In temperate parts of the world, you have to have your compost piles minimum 3 foot tall, 3 foot in diameter, or you can't maintain temperature through a cold night. When we're in Nebraska and we're composting through the middle of a -72-degree temperature period, you better have at least 10 foot across, about 8 foot high pile, or you're not maintaining temperatures through that cold winter period. And you better cover the pile, because if you have snowfall on top of your pile and it's nice and hot, 5 feet of snow melts into your pile. Great, that tends to wipe out your temperature. So some of the lessons I learned along the way of what not to do. You don't have to fuss about that here in the tropic. So you can have pretty small sized piles and compost away right underneath your tree. So as soon as you're done composting, you just spread it around the tree. You do need a thermometer. Well, we'll talk about all of this.
Is it better to, like we're gonna do a sample, yeah? Because and we got some wood chips and some leaf cuttings, and then also some straw with the manure. And is it better to keep it covered?
And I've worked a lot with Chuck Warner over on Maui. I find that you have to cover everything in this part of the world because it gets totally waterlogged. When you get 3 inches of rain in half an hour, everything's totally waterlogged. So I find you have to keep it under cover.
Yeah.
You have to have it covered, and then your compost pile has to be under cover because, yes, how did Chuck learn not to leave it uncovered? Now, you want to see some
I guess my
red
next question would be like, if I got everything ready, could we do that in the morning?
Tomorrow morning? Sure.
Yeah.
Yeah, so we can put a compost pile tomorrow,
Yeah.
and I'll briefly talk about the recipe and
Right.
how you put this
I
together,
don't want to mess up your, what you call that,
because we're already behind. So, this is my background. Why is it that I get to stand up front here and talk to you about all of this stuff? Because I've been working in this area since a long time. Last week was my 60th birthday. And so I've been working, when I was 6 years old, my dad took me into the laboratory. He was a professor of veterinary medicine at the University of Minnesota. He sat me down at a microscope when I was 6 and said, count the E. coli. So I've been doing this for a long time. I've been working on compost tea since about 1976. So my background, there it is. I grew up in Minnesota. You can hear the accent in my voice every once in a while. It sneaks in there for sure. And don't you know? You betcha. They sneak out there every once in a while. So I grew up, went to St. Olaf College as an undergraduate, a double major in biology and chemistry. So I have both backgrounds. Yes, I can run an NMR, nuclear magnetic resonance machine. What fun. I love those things. They're great toys. Guys, you like tractors and big trucks? I like mass specks. Those are fun. Okay, a little weird. So, grew up there. How many of you remember the Golden Girls? Remember that television show? You remember Rose? She was always talking about St. Olaf and the wild and crazy things we did there. They're all true. And how do I know? Because I did them. Yes, jumping into the creek in the middle of the winter when it's minus 50 degrees, just because we wanted to know how fast you would freeze to death. Yes, we're not to do that quite quickly. So double major, then went on to graduate school at Texas A&M, where I worked in the world of marine microbiology. Doing the work in marine microbiology, the question that I was given when I walked in the door was, why are the oysters in Galveston Bay not growing? There was an oyster industry in Galveston. And the problem was that they had tried every chemical known to man to put into those oyster beds. They put fertilizers, and they put hormones, and they put enzymes, and they put everything they possibly could. And finally somebody said, well, maybe it's a problem with the biology. Don't oysters have like stuff in their digestive system? So they came to my major professor, and just a few weeks after I started at Texas A&M, and so my major professor said, so why do you take this one on? Okay, great. We're gonna look at the biology in the digestive systems of oysters. So how do you look at biology in the digestive systems of oysters? Well, the classic microbiological method was to do plate counts. So using a method that had been developed by Robert Koch, the father of modern medicine, back in 1750, plate counts. And the idea with a plate count, I kind of briefly clued you in on this one before, but let's make sure y'all understand this at this point. So a plate count, nice little petri dish, petri, named after the guy who made the first petri dish. So it used to be glass, today it's plastic, and it comes sterile. So that petri dish, it has a lid typically on it. So when we put that lid on it, it protects the contents of that plate from getting anything on it. But because it's a lid on a nice plastic material, it's like a swan neck tube. You cannot get anything from outside here to come up under the lid, over the edge, and into the plate. So it maintains no contaminants getting into the plate, and oxygen cannot move into the plate. No exchange of atmosphere from inside the plate to outside. Typically what we're going to do is we're going to put a medium. So we're going to pour a nice thin layer of medium into that plate. What's a medium? Well, it's a specific set of foods. When we were looking at total bacteria in the digestive systems of these oysters, total bacterial counts, we used tryptone soy agar. How many microorganisms are you actually going to be able to grow on tryptone soy agar? What's tryptone? It's one kind of protein. One protein. How many bacteria in an oyster's digestive system grow on tryptophan? Think about soil. If we're doing total bacteria in soil, how many bacteria in soil utilize tryptophan? Soy. How many kinds of food resources in a soybean? So take a soybean, throw it in boiling water, boil the living daylights out of it, and that soup that you've made contains about 12 proteins, a couple of carbohydrates, something like that. Agar is just a solidifying agent. So you can heat it up, add these foods into it, get a nice liquid, pour it into your plate, cools down, and now at room temperature, it's a solid. So it's just a solidifying agent. It's not food for anybody. How much agar do you find in soil? Seaweed in soil. So not a food resource. So now think of all the different kinds of bacteria that exist in soil, or all the different bacteria that can be in the water column that an oyster is filter feeding. Are we providing foods to grow all of the different organisms that could exist in the soil? And if you laugh, how many food resources would be in a typical gram or cup of your soil? Think of every leaf that's ever fallen to the surface of that soil. How many different kinds of foods are there in a leaf? 100,000? Okay, let's have 5 or 6 different kinds of plant material falling into that soil. How many kinds of foods in your soil? 500,000? So how much of that actual set of bacteria that are present can we grow on a plate where we're putting in 1/1,000,000th of the number of typical foods that might be in existence in your soil or in your oyster's digestive system? See the problem? We're not even coming close to providing all of the foods that may be present in any soil or any digestive system. So what are we missing when we use a plate count? Now, we were looking at the biology in the digestive system of the oyster. So here we are. That's the liquid from inside a digestive system of an oyster. We've got some lactobacillus. Isn't that cute? There you go. You can actually see lactobacillus and what it should look like. Well, here's a different species of lactobacillus. Look at the fact that this one's only this long, that one's that long. Well, we've got another lactobacillus. That's a different species. And we've got a third species. And there's another one. This one is actually sitting here with a short one, a short one, a short one, but they chain up like this. Every one of these is a different species of lactobacillus. We've got some flagellates. Look at them running around eating bacteria. They're having a grand old time. We got some yeasts in there. See the guys that are buddy? Those are yeasts. We got some individual bacteria. Which species? We've got a bunch of them, lots of different sizes and stuff. So how many bacteria do we have in here? And how many species? We've got like 1,000 bacteria and 12 different species just in this teeny tiny bit of juice coming out of an oyster's digestive system. Now we plated this material on tryptophan soy agar. How many species? 2. A lot and a big 2. See where, see the discrepancy? What would your conclusion be about what's going on in the digestive system of an oyster using this approach? We don't have very many bacteria. It's pretty sterile in there, right? Wrong. So I started doing this kind of work because I was already staring through. I was used to microscopes, and I was looking at this stuff through the microscope. And I go back to my major professor and say, "I got a problem here. How do you explain this?" And he was like, "Well, you did something wrong." Okay, how do you prove that to somebody who doesn't want to hear and doesn't want to listen? So after 1,000 samples, going back to him with a number of species that he could identify, all the pictures, and saying, this methodology just does not work. Think about the fact that we are restricting the number of species because we're restricting the number of foods in here. Well, once we've spread our oyster digestive fluid on that petri dish, what are we now going to do with that petri dish? We're going to put it in the oven at room temperature. 72 degrees Fahrenheit. What's the temperature of the water in Galveston Bay?
55, 60.
Yeah, if you're lucky, it's 60. You know, so not exactly fun most of the time. Swimming in Galveston Bay is a little cold. So, are we even matching the organisms that we're actually growing in the digestive system on that petri dish? And then think about on that petri dish, we're dealing with a moisture content that's about 15% moisture. What's the moisture content inside the oyster's digestive system?
100%.
See where there's a problem? And then think about the fact that each one of these bacteria, when they started growing, it was a single bacterium or a single colony forming unit, maybe 2 bacteria, maybe 3. But along in the course of 24 hours, that 1 or 2 or 3 bacteria started to grow. 1 becomes 2, 2 becomes 4, 4 becomes 16, 16 becomes 84, and on and on and on. As these bacteria are growing, what are they doing to the oxygen in that little atmosphere that's on the surface? It's going anaerobic. How fast does that atmosphere go anaerobic?
15 minutes.
Yeah, so here we are. We went anaerobic along about, what, 30 minutes? For like maybe 60 minutes? So what kind of organisms are actually growing on that petri dish? Aerobic organisms? No way. What are we actually looking at when we do petri dishes like this? Pathogens. Because pathogens require reduced oxygen in order to grow. So this in no way represents the organisms in the digestive system of an oyster, because if you go anaerobic in the digestive system of that oyster, it's a dead oyster. So, started showing my major professor what the problem was with the classic method of assessing biology and environmental samples. So when you start looking at some of the silly stuff that's in the scientific literature and how people try to pull the wool over people's eyes, when they say, oh, our pesticides don't kill anything good in the soil, they are not using direct chemical determinations, they're using petri dishes. What are they actually telling you about? So this is a method that was developed to grow pathogens. Robert Koch, Louis Pasteur developed this in order to demonstrate human pathogens in different kinds of materials. And it grows pathogens, great. But do not even imagine for a second that it's telling you anything about total bacteria or total fungi or anything else. Interpret this information correctly. So there you are, you've got these, this is written up, all the problems. You know, when we're doing total fungi, what's the medium that they use? Potato. Dextrose agar. What's potato? Starch. Take a potato, peel the potato, throw the potato in water, boil the hell out of it, and you've got a starch solution. How much starch appears in soil? Not. It doesn't really. Well, maybe if you broke up a potato or, a taro plant. Dextrose, what's dextrose? Sugar. White sugar. One specific kind of white sugar. Not the left-hand sugar, but just the right-hand sugar. So how many fungal species will grow on this stuff? Yeah, there is no way you're learning anything about the fungi in the soil. You're learning about the pathogens. And you might not even be learning everything about the pathogens. Some of the other methods, so in working on my master's degree, looking at some other ways to assess the biology inside the digestive systems of those oysters. We can enzyme assays where we can ask questions about how many bacteria utilize this one particular kind of cellulose. And how many different kinds of cellulose are there on this planet? A couple 100,000? So what are we finding out with one single enzyme assay? You know, different kinds of sugars. How many kinds of sugars are on this planet? A couple million? You know, and it just drives me crazy that we would use these kinds of methods to tell us about all of the activity or all of the sets of organisms that are doing things in soil. CO2 evolution. Carbon dioxide is released when you have aerobic decomposition. But what happens when you go anaerobic? So we're going to put our compost in this jar, and we're going to measure the amount of CO2. What if your compost is anaerobic? You're actually taking carbon and tying it up as methane. You're converting it into something other than CO2. So we have a negative CO2 production. How do you interpret that? You see some problems with most of these methods. You have to understand what you're doing when you're trying to interpret any of these methodologies. If you want to get about a 45-minute lecture, just ask me about this one. I'm not going to go into it because it's just, oh gosh, silly. So, you know, when we want to use plate counts, it's to understand how many E. coli do we have in our soil. We know how to grow E. coli very, very well. We know how to grow Salmonella, Shigella, Pasteurella, all the human pathogens. Boy, we can grow them. And so that's what plate counts should be used for, but not for an assessment of totals. So life in oyster digestive systems or when plate counts are pointless. So looking through the digestive oyster system, what we found was that whenever we were dealing with healthy oysters, you would always find a specific bacterium in the digestive system of that oyster. You would always find Cristospira. And if you don't have this organism in the digestive system of your oyster, the oyster will not gain weight at all. Period. Full stop. It sits there and looks at you. So when we went out with inocula of Cristospira, so that was one thing I did for my master's degree, was to develop a growth medium in the laboratory for this particular bacterium that had never been cultured before. And so we grew up high numbers of Cristospira, released those into the oyster beds. The oysters all started growing. It was amazing. Come back 3 days later, Cristospira's all gone. Oysters aren't growing. What happened? We started to correlate every time we put the Cristospira out and came back a couple days later and there was no Cristospira. It's every time they were releasing pollutants from the Navy shipyards. Every time they took one of the boats out of the water and did the biofilm, all that garbage, the
Anti-fouling?
Pardon?
Anti-fouling?
Anti-fouling materials or removing the biofilm from the bottom of the ships. Really nasty compounds that they use. So it's really strong acids, strong bases to remove that the organisms growing on the bottom of the boat. And then they proceed to paint the bottom of the boat with lead-based paint. So downstream, on comes, and so every time I'm looking at these oysters after a couple days, and inside the oyster is this gray film of lead-based paint inside the oyster. And so I'm going out collecting oysters, and people are in Galveston Bay walking out into the water, pick up the oyster, shuck it open, and slurp it down. And I'm going, you really shouldn't do that. Really? That's not a good idea. I've been doing that my whole life. Yeah, now we know why Texans are the way they are. Man, it's fun. See, even he said it's true. Especially the ones that are drooling out the sun. That's why they dance the way they do in Texas. So, you know, think where does the Bush family come from? Where do they vacation every summer? And they're out there in the bay and they're shucking down those oysters. See, this is what environmental pollutants do for you. So it was, well,
Manchurian candidate.
it was just
Yeah,
impossible trying to keep those oysters growing. There is no oyster industry in Galveston Bay anymore, but in a lot of other parts of the world, they use this medium for growing Christus viola. They put it back into the oyster beds so the oysters will keep growing. We have to keep putting this bacterial inoculum back in there. What's that really telling you about the health of the oysters? I'm not eating them. Because what kind of toxic materials are in there? So, looking at how, not even true. Colorado State University. We started working with a comparison of organisms, biology, and all of these different systems. So for my PhD at Colorado State University, so that was my master's at Texas A&M, now we're on to my PhD at Colorado State University. I jumped from marine microbiology because I realized we couldn't get any money to actually stay alive in the world of marine microbiology. So started in the world of soil microbiology. Did a determination of active organisms in soil. So came up with an assay that we could determine active biology in soil. And there's a particular stain that is only taken up by active growing organisms. This is fluorescein. Diacetate, so there's the D. It's not the Food and Drug Administration. This particular stain is not fluorescent when it's tied up with these two acetate groups. So when you're looking at fluorescein diacetate, it's a fairly complex molecule. And here on this carbon, there are two acetate groups. So carbon-carbon bond, bonded to another carbon. Each one of these little corners is a carbon molecule. And then these R's are some 5 or 6 chain long carbons. So this is fluorescein diacetate. Once an organism cleaves off these two acetate groups, then you get it looks like a benzene ring, and it acts to absorb light at one wavelength, and it releases light at a longer wavelength. It becomes fluorescent. So this compound has to be actively transported across the cell membrane, which means you have to have a living, active, growing organism to take this up into the cell. It has to be pulled in against concentration gradients, so it takes energy. Once it's within the cell, your organism has to be active and alive, producing esterases to break those two acetate groups off. It's food for these organisms. And then that releases the fluorescent compound. So we stain organisms for 3 minutes, no longer. And we assess the organisms, the bacteria, the fungi, the protozoa, the nematodes that are so metabolically active. They'll take this up and concentrate it to the 9 micromolar concentration within the cell, and now you will see that cell as fluorescent. So it's only it's the marathon runners in your soil. It's the guys that are really cooking, really active. And so we developed that methodology. So I began to do this work in all of these different ecosystems, looking at active bacteria, total bacteria, active fungi, total fungi, active protozoa, total protozoa, nematodes. Somebody else did microarthropods, somebody else did plant assessments. And what we discovered is that I've arranged these ecosystems we were working with from least productive systems based on total plant production, nothing on food made, total plant production. The least productive system is irrigated wheat. Had the poorest food web. This is strictly bacterial dominated. Very fragile, very easy to lose all your production in the system with. Just the slightest thing going wrong. Dry land wheat, a bit more productive. Fewer weeds in the system. Short grass prairie. So I don't know how many of you have gone out to Colorado when you're in the Front Range. That land to the east of the Front Range, short grass prairie. We're in the rain shadow of the Colorado Rockies. So not very productive systems, relatively speaking, but more productive than either of the conventional agricultural systems. Move into the tall grass prairie. Kansa National Grasslands, going out there and measuring the productivity, the biology in the soil. It's always fun when you're out in the tall grass prairie, because how tall is the tall grass? It's about, yeah, the year I was out there, 12 feet tall. Wow. So you're out there in the tall grass, and taking all these measurements, and you realize the sun's starting to go down, and you stand up and you go, which way's home? Hey guys, someone, hey. And you realize that, where you walk is all, the grass is all sprung up. And you really worry about how you're going to get home. There's a few grim moments out in the tall grass prairie. Obviously I found my way home, but you learn to come along with a compass. Because you always want to, we're going to go east. And so when we want to get home, we're going to go west. So you bring that along. That was one of the funner experiences of my life. She's lost, and will she ever find her way? How many miles is it that way to civilization? About 150. So I picked the right direction eventually. High mountain meadows, very productive systems. They run cattle and sheep. And so when we're looking at the weight gain of the animals out on those meadows, that's a measure of how productive those systems are. So we're adding in the production that's removed by the cows and the sheep, as well as the actual plant material that's being produced and staying in those meadows. And then of course the most productive ecosystem, the lodgepole pine forest. On an annual basis, not all that large an increment of bark or wood going into the trees. 80-year-old trees are only about that big around DBH. Those trees out there, how old are they? 15 years. So huge difference in water. But there's a huge amount of understory in these lodgepole pine systems. There's a lot of ephemeral grasses and herbs, different kinds of plants come and go, and you gotta add in all of that productivity. So this is arranged in the order of production. How much plant material is being produced? And this goes from strictly bacteria. There's a little bit of fungi here. There's a little bit more fungi here. There's a lot more fungi here, almost an equal biomass of fungi to bacteria in the tall grass prairie. High mountain meadows actually slightly on the fungal side. And in the lodgepole pine system, massive amounts of fungi in that soil. The first time ever documented are in the publications that come out of my PhD work at Colorado State University. So the first time we're really documenting that it is the organisms in the soil that set the stage for the plants that are growing there. When we take materials from these systems and put them into those systems, no problem with decomposition. But when you take materials from here and put them in an earlier successional system, that plant material sits and looks like here. We have some pictures where some of those plant materials that were put in, some of the grasses, some of the woody material from here, put into those systems are still sitting there untouched. And this was work that was done back in 1985. And they're still sitting there. Here we are at 2012. Because there's no organisms in those systems to decompose that material. The fungi don't exist, they are not present to decompose that material. Take these materials, no problem. We got the organisms. So look at the effect of the biology. First time ever starting to document this in the work that we did at Colorado State University. We discovered that the most rapid rates of decomposition actually occur in the winter under the snow. So in all of these systems, as soon as we have a nice deep, like about a 6-inch layer of soil on the surface, that's when we see the most rapid rates of decomposition. Now when I published that, there were a lot of people in the ecological world that were like, man, this woman is so crazy. She has no idea what she's talking about. We're going to prove her wrong. So they get money, they go out there, and then a couple years later they come out with a publication that says, we discovered that the most rapid rates of decomposition on the planet are in winter under the snow. Thanks. Yeah. Academics, are they nice people? So it was kind of fun. It was an eye-opener. The ecological monograph comes from the work we did at Colorado State University, where we established what organisms are responsible for which parts of nutrient cycling. So Ingham et al. Was published back in 1986. It's an ecological monograph because in this monograph, what we had to have was, first of all, here's the control, sterile. And then we put a wheat seedling in there, so there's soil in the bottom of our microcosm. Isn't that a cool name? We were playing with microcosms. What was this really? Pint-sized mason jar. Soil in the bottom, sterile, put a wheat seedling in there, and see what happens. Anyone want to bet on how long that wheat seedling survived in sterile soil? About 3 days. So then the next treatment was just bacteria in the soil, and the wheat seedling survived for about a day and a half. You put bacteria, and only bacteria in the root system of wheat seedling, yeah, no nutrient cycling. Killed it really fast. Just fungi. And then the next one with just protozoa in the system, and the next one with just bacterial-feeding nematodes, and the next one with just fungal-feeding nematodes, and the next one with predatory nematodes, and the next one with root eaters. Yeah, how long did the plant stay alive with root eaters in the root zone? It didn't even make it to the end of the day. So now we started adding in bacteria plus protozoa, bacteria plus fungi, bacteria, all of so we just started building the complexity within these different treatments, demonstrating that the only time the plants stay alive is when you have bacteria and a predator in the system, fungi and a fungal predator in the system. The best, most rapid growth of all of the wheat seedlings was where we had bacteria and fungi and protozoa and nematodes, good kind of nematodes in the system, showing the uptake of nitrogen, phosphorus into the grass, into the blades, clearly demonstrating that it is that whole food web that does nutrient cycling. Now, I do like to point out that I am not this Ingham. This is my husband. So my husband and I work together on our PhDs and on our postdoc, working out this system. I hope all your plant material is covered.
No.
Oops. Down forest. You gotta like tropical systems for that. So this is my husband's PhD work from Colorado State University. I helped do the bacteria, the fungi, and the protozoa in the system. He did the nematodes and a lot of the plant system. I am the third author on this paper. I'm not the first. And you will get some people who will like to say, well, Elaine likes to claim that this ecological monograph is hers. No, I'm not claiming that. But it is on my list of publications because I'm the third author on this paper. My husband is the first author. So I was right in there having fun with that system. So don't want to get away with it. So then I started while I was at, 5 more minutes. Okay, I think I can go really fast through this. At Oregon State University, I started working in the Department of Botany and Plant Pathology and in forest science. Not real smart. Split between 2 different colleges. They both thought they owned me. It was hard to keep them all happy with me. So when I had been at Oregon State University for about 10 years, I started an intergovernmental personnel agreement with the Corvallis EPA, and I went to work for the biotech team at the US EPA. The question that we were asked was, when we look at genetically engineered microorganisms, is there an ecological effect of a genetically engineered microorganism when it's put out into the system? So the first 14 species that we worked with were all human pathogens. And all these human pathogens were engineered where we added antibiotic production to those bacteria. This is an energetic drain on that bacterium. So when you look at whether this bacterium survives in soil or not, this antibiotic production meant it did not survive very well at all. How long does a pathogenic bacteria survive in soil to begin with? About 24 hours. So instead of surviving for 24 hours, this thing survived for 18. So on the basis of these first 14 species of bacteria, USDA APHIS, the Animal and Plant Health Inspection Service, using this data, said that genetically engineered organisms, right, we only worked on 14 species of bacteria. How does that become genetically engineered organisms? Where's the data, guys? If they had made a genetically engineered microorganisms, but even that's not valid here. Genetically engineered organisms are of no greater risk than the parent. And I argued for a long time that you can't say that. We don't have the data. You cannot take that interpretation from the work that we were doing. 14 species, and you're going to decide all genetically engineered organisms are of no greater risk than the parent? I don't think you can do this. But who was really paying the bill here? Okay. I had a graduate student, Mike Holmes, who said, this is just not right. We got to do something. So for his PhD work, what he decided was that he was going to work on a genetically engineered microorganism that actually came from soil. It would live, it would survive in soil. Because that's a huge problem with this, is that none of these organisms live in soil. They don't survive. So how can you really say what the ecological effect is going to be if they can't survive in soil to begin with? So he went out and he found a bacterium, Klebsiella planticola. This is a normal soil organism. It's found in the root systems of all plants. Every place on the planet where you look, you will find this bacterium. That's the parent. It decomposes exudates from all root systems. It decomposes leaf material from every plant we've ever looked at. It is a truly universal microorganism. They took the genetic material from another bacterium for alcohol production and put that into that bacterium. So now we have a Klebsiella planticola that makes alcohol. Well, what's wrong with alcohol production? No, he had a committee that looked at this and said, okay, we're going to prove that these genetically engineered organisms aren't going to harm your plant. Well, what were they trying to use this Klebsiella planticola engineered to produce alcohol for? The idea was to stop fuel burning. Stop fuel burning. At that time, huge problem in Oregon. Every August, September, you could not go outside because the air was so heavy and thick with smoke from burning all the grass fields. Oregon produces 60% of the world's grass seed. And so when you have to burn the fields to get rid of all the residues, you shouldn't have to do that. Why would you burn the residues off your field? Well, because it's going to grow a bunch of pathogens. And if you get a good healthy food web out there, you don't have to do that. But never mind. Instead of field burning, you would be able to break up all of those residues, put them in a big bucket on the farm, inoculate this genetically engineered microorganism, seal the lid, and in about 2 weeks, you could open the spigot at the bottom of that bucket, and out would come a material that's about 17% alcohol. You gonna drink this? This is like vodka. This is 34 proof. How many of you drink vodka? What's it made of? Potatoes that are fermented. So let's do it with grass seed material. 17% alcohol. You could distill it, you could make a lot of money. So isn't this a win-win situation? Stop field burning so you can actually breathe in the Willamette Valley in the fall. And you're making a product that the growers can sell. And even better, you take the sludge that's left in the bottom of that bucket. How many of you ever made beer or wine? Think of all that sludge stuff on the bottom of the bucket. This has got concentrated all the nitrogen, phosphorus, sulfur, magnesium, calcium, sodium, potassium, the whole shebang. So you can take this material and put it out on your field as an organic fertilizer. Win, win, win. There's no waste product here. Okay, but when you're inoculating, when you put this material out on your soil,
Pathogens.
you're inoculating, no, there could be some pathogens in here, yes, but how about a genetically engineered microorganism that makes alcohol? The alcohol doesn't kill Klebsiella planticola, sorry, it doesn't touch it. It is not sensitive to that material at all. So, what happens when you put that material out on the soil around plants? And that's what my graduate student did, that's what my homes did. So again, microcosmos, this is kind of fun. And 3 different treatments. All 3 treatments had the same amount of soil from the same place, sieved exactly the same way, randomly put in the units, and randomly pick the units. Into the first set of treatments, they just put in water. Put a wheat seedling in there, started growing the wheat seedling. So there's our control. How does that wheat seedling grow in a system that's just got the organisms in the soil? To the next one, in the same amount of water, they put the parent Klebsiella planticola. Put the wheat seedling in there, let it go in the greenhouse. Into the third one, they put the genetically engineered microorganism. You gotta appreciate the EPA, genetically engineered microorganism. It's a gem. It's gotta be a treasure, it's gotta be good, it's a gem, right? Same amount of water. And now there's the wheat seedling in there. Put it in the greenhouse, let it start growing. So that was Monday morning that he put this experiment together. By Tuesday morning, it's all up and running. Everything's in the greenhouse, looking good. It calls me up Saturday morning. They've been planning on going out to a fair or the Renaissance Fair or something like that. And so he'd come in early to water his plants and do the mixing. And 6 a.m. he calls me up. Dr. No, oh my God, it's just terrible. I'm gonna flunk it. It's ruined. It's all gonna be dead. It's just terrible. What am I supposed to do? I'm gonna have to go. I'm not gonna be able to graduate. Calm down. What is the problem? What, did the lab go up in smoke? Did it burn down? What's going on? Really upset because a whole bunch of the plants are dead. Just flat out dead. And he's gonna have to start over. It took him 6 months to put this experiment together. All the testing, all the things that have to happen, he's gonna have to start over again, right? That's what he's saying. And I was like, so calm down. Start taking these units out of the greenhouse and start putting them in the treatments because they were just randomly mixed together. So he starts pulling them out, and all of these plants, after less than a week of growth, all of these plants have grown significantly. So wheat plants grow when you provide them lots of water and good sunlight. So great, there's my control. These plants, where the parent Klebsiella had been put into the system, were trying to grow out of the microcosm. Way more tillers, way more biomass of plant material. What's the effect of the parent Klebsiella on the growth of wheat? Would you all agree with me that it's beneficial? So this is helping your plant. Every single plant in these, where we had added a genetically engineered microorganism, all of those plants were green slime on the surface of the soil. Genetically engineered microorganisms are of no greater risk than the parent. Here's the experiment that puts the lie to that. So he put this experiment, he wrote it up. We took it to the head of the EPA, and the response from Bob was, you made a mistake. After we repeat the experiment. Do it over, you messed up. We were 2 weeks from the release of this bacterium in a field study. What would happen if this genetically engineered microorganism got released out to the real world? We would lose, that's just a conclusion that is inescapable given what this bacterium can grow in the root system of all plants. It decomposes plant material from all plants. If we release this organism out to the real world, we would lose terrestrial plants. Think that might impact your life? No tomatoes, no avocados, no corn, no wheat, no barley, no grass, just think of terrestrial plants. Would that impact your life? Kind of scary, isn't it? What, terrestrial plant, it grows in dry soil. It grows in places where the soil gets dry. Wetlands, it's waterlogged for a certain period of the year. Riparian, waterlogged for at least 40 days. Terrestrial is everything else. So we would all have to live on rice for the rest of our lives. And seaweed. I'm real thrilled for that world. So Mike and I agitated a lot to prevent that experiment from being done. We did stop them because they thought, the head of the EPA finally said, well, maybe, okay, we won't do it. If you go back and ask him today about that experiment and were they going to release this bacterium out to the real world, he will disavow any knowledge of that experiment for anything happening. So I took this data to the United Nations Biosafety Protocol meeting in Madrid and gave this information at the end of the first day of those meetings. The scientists from 12 different countries around the world had gotten together before this meeting and come to the conclusion that genetically engineered organisms were of no greater risk than the parent, and there was lots of data to prove that. And I kept pointing out to everybody at the meeting, that's based on 14 bacterial species. They have no data for plants. They have no data for fish. They have no data for anything. That policy is based on bacteria. And maybe there's a problem with that. At the end of the first day, after U.S. government representative stands up and says, don't worry, be happy, there's no problem with genetically engineered organisms. We would never do anything to kill give any problems to the people who are our clients. And I'm back there going, wait, you never heard of tobacco companies? You never heard of pesticide companies? How come you have to withdraw all these pesticides from the market? Who let those out? Killing people. So, come on, guys. Second to last speaker on that day, the NGO, so I was there with a non-governmental organization, asked me to stand up and give this talk, basically. Into my talk, you could have heard a pin drop. All of a sudden, it went from, we can play all week long, we can just go out and have a good time here in Madrid, it's a wonderful city, to, we're actually going to have to do something. We're going to have to write a biosafety protocol. Of course, as soon as I got done speaking, who pops his hand up? The U.S. representative, and he stands up and he says, we're not talking pink elephants here. We're talking real world, and this is just a laboratory experiment. Pay no attention to the crazy person behind the curtain. And I stood up and I said, I got 30 seconds to respond. This is completely real world. This was real soil. These are real wheat plants. Everything about this mimics what goes on in the real world. And so, went out to dinner last night, and I got so many people coming up giving me drinks. Yes, you've really changed this. You've altered the whole. We do have a biosafety protocol at this point. Of course, the problem is getting them to actually apply the law. Yeah, go out and make them pay the fines for seeking genetically engineered organisms all over the world. But we at least have a biosafety protocol. Of course, I get done with my, all these cheers and everything's wonderful now. We got a biosafety protocol. Went back to Oregon State University, walked into the door, into my department. My department head comes up and says, "You in my office now." Okay. Walk into the office, sit down. My department head says, "We have a real question about the quality of your research. Your methods are questionable. You don't know how to do good science." I've been a professor at Oregon State University for 15 years. I have over 100 publications in scientific literature. No one has ever questioned my methods. I have the audacity to go up against Monsanto, and all of a sudden, I don't know how to do research. So I held out for a couple more years at Oregon State University, but I could finally read the handwriting on the wall, and knew I wasn't going to survive in the Oregon State University system. One of the past presidents of the university wrote a letter to all of the faculty members in the university that said, if you, as a faculty member here at Oregon State University, if you don't believe in biotech, if you don't believe in genetically engineered organism, you probably don't belong at this university.
That's why I didn't go to
Because, who's in control there? Jeez. They made it impossible for me to stay. So I started Soil Food Web. And so, as an experiment, you've got this in your data, in your handout. So they made it impossible for me to stay, so I started Soil Food Web as a result of realizing that I wasn't going to last that much longer. My department at Oregon State University, botany and plant pathology. I was bringing in about $2 million worth of research funds every year, and Monsanto was contributing $75 million to that department. So, of course, what's my department head going to do? Who are you going to piss off? $2 million versus 75. Who's winning? Universities are big business. Sorry, they're big business because they got a whole lot of salaries to pay. So when you go up against somebody who's contributing that much money, geez. So yeah, I started Soil Food Web and started talking to people all over the world about this process of these basic talks that I'm giving to you now, and looking at effects like this all over the world. And we will be talking about more experiments like this. One application of compost tea. Without compost tea, look at the difference. So this is about 3 days after they sprayed the first application of compost tea. Look at the difference. Which kind of plant do you want to be eating? So disease suppression, building structure in the soil, getting nutrient cycling going, very rapid changes can happen because you get that biology in the soil. So I can give you lots and lots and lots of examples. So 1996 we started Soil Food Web. At this point I'm kind of backing away from Soil Food Web because it's a business. We got it started. You need samples done, send them into Soil Food Web. And they don't really appreciate having a research scientist around constantly saying, we ought to do this experiment. We don't have the money to do that, Elaine. We should do this experiment. Okay, so hard to work with growers and get replicated scientific trials done. So when Mark Smallwood asked me if I knew anybody that would like to take the chief scientist position at Rodale Institute, I thought about it for a day and I emailed him back and said, well, what about me? And he said he was dumbstruck when I said that back to him. His response to me was, really? Yeah, what kind of deal you got on them? So at Rodale Institute, we're going to get the replicated scientific trials done. We have 333 acres at Rodale, and half of all of our fields now are having either compost tea, compost extract, or compost put on them. The other half not getting those applications. So coming the end of this growing season, next growing season, the growing season after, in the world of replicated science, not only do you have to replicate the fields, but you have to replicate over 3 years at least. So in about 2.5 more years, we should have a massively large paper coming out where we're showing these kinds of results. So that's where we're at, and that's where I'm at Rodale now. Chief scientist.
So, Elaine, is that why now Hawaii is the GMO seed incubator? Because if they really mess up,